Aloha, and thank you for joining this panel discussion. My name is Aurelia Gonzalez, and over the last 2 years, I've been trying to document the aquatic biodiversity of Pearl Harbor. In an attempt to accomplish this biological feat, I came across a comparative study between sequencing animal DNA from a water sample and capturing animals through traditional fishing practices and survey equipment, such as underwater cameras and cast netting. This project was in partnership with Navy Natural Resources and the Zhang Lab in the NREM department at UH, as well as Mark Renshaw from Oceanic Institute, who carried out the genetic processing for all of our eDNA samples. The second half of this project couldn't have been done without Glenn Higashi and Neil Hazama from Division of Aquatic Resources. They provided their years of biological experience, their equipment, boat, and local knowledge. Estuaries are one of the most important ecosystems recognized across the world as supporting the economy and the environment. Our ability to monitor species in estuaries is of particular importance as these waterways are some of the most impacted habitats due to habitat loss, species introductions, and poor water quality due to anthropogenic disturbance and alteration. In order to support and maintain a balanced system for estuaries and the ecosystem services they provide, it is critical for biologists and natural resource managers to monitor aquatic species in these complex habitats. As recent as the early 2000s, aquatic scientists have been examining modern applications of detecting environmental DNA across freshwater systems in order to identify the presence of species and define community assemblages. eDNA surveying consists of a soil or water sample taken from an environment where DNA deposited from organisms is captured and sequenced for genetic analysis. Specific species of interest, such as a rare and invasive species, to identifying entire compositions of taxonomic groups, can be identified in a single water sample. The idea is to create a rapid assessment of species distributions with data that provides more spatial and temporal resolution, is easier to acquire, and is taxonomically reliable. This biodiversity study surveyed 5 stream mouths of the Pearl Harbor estuary, quarterly for 2 years. We used 4 traditional sampling methods that included an underwater camera, cast netting, minnow trapping, and angling. Along the same transect line for each stream, and on the same day, we took multi-depth eDNA water samples. We used a technique that is referred to as metagenomics, where we targeted DNA using 4 different genetic primers to capture a wider array of species. For analyzing the strengths and weaknesses of a mixed method approach, we standardized our sampling effort at each site. We made sure to record our fishing effort and identify our area of coverage for each gear type. We controlled for DNA contamination by sanitizing our equipment, including field and PCR control samples, and having a mock community sample in our lab procedures. The data is made up of presence-absence accounts from each equipment method, stream site, location in the water column, and sampling date. We can then derive alpha and beta diversities and construct percent agreements, detection probabilities, and find covariates of detection. The hardest part about combining data sets is the quality and control over the scientific and common names used for species. You must backcheck the consistency and nomenclature across your data sets. There are misidentifications historically, culturally, within genetic libraries used. There are differences between primers. And there's a lack of local reference sequences, which puts in question true positives. Biologists, local experts, and land managers need to come together to review the data for false positives and likelihoods of species occurrence. This reveals improvements for the sequencing process and possible contamination events. So far, our preliminary data has indicated the need to implement a mixed methods approach to surveying estuarine species. Both eDNA surveying and all 4 traditional sampling methods have detected unique species. We are also finding species that are historically missed in traditional surveying practices, but are being detected by eDNA methods. Cross-method comparisons between traditional and eDNA surveying have allowed scientists to assess eDNA as a surveying practice. Before closing out, I would like to make a special shout out to Mariah Baker. She was our biological technician who was extremely dedicated and necessary for making this project happen. She helped in the field collections, data entry, and species identification for this project. Mahalo for coming out and listening to this discussion today. Aloha.